begin, I would like to thank HFSP for believing that 3 groups that think that chicken and jellyfish should be studied in the same sentence is a good idea. And these 3 groups is the group of Annemiek Cornelissen in France, and her postdocs Laureline Joulier and Thibaut Chassereau, who is doing experiments in jellyfish, Liz Jones in Belgium, and Cara Trivet, who are doing experiments in vascular remodeling. And last, my group from the United States, and Paheli, Zimbo, Aaron, and Alex, who are doing the theory and computation for physical networks. So the theme, let's say, is biological transport networks. Now, all of you here who have talked, you have implicitly or explicitly talked about transport, because without transport systems, the evolution of complexity in biology would just be impossible, because essentially transport allows division of labor and specialization, right? And these are there on the slide. You can see some systems where that is relevant. Now, in this talk, you're going to hear a lot about fluid flow networks, and the question that we're going to be asking is how these networks are being built, right? Now, there is something in, let's say, fluvial geomorphology called the morphogenetic trinity, the morphodynamic trinity. And that basically says that we have geometry, and in our case, in biology, that would be form. Specifically, in our case, that would be the structure of the vascular network, like the one you see over there. OK, that's a bad pointer. I'm not going to use it again. Now, that form informs a morphogenetic field, which is the function. And in our case, that would be flow, mechanical signaling, morphogens, and so on and so forth. And that, in turn, will inform some growth rule, which is going to be development, and in our case could be tip growth or remodeling. Now, a very big part that has been, I guess, severely understudied is dynamics in the morphogenetic field, and this is what this talk is going to be about. Eventually, this project was going to be about development. There was a poster yesterday by Annemiek which talks about development. Today, I'm not going to talk that much about development. Now, information is globally integrated to transport in this trinity, so the whole organism, or organ at least, needs to be considered to understand what's going on. Now, so my slides are not displaying well at all, although we tested them, so I apologize for that. So the 2 heroes of the story here is, as I said, the avian yolk sac and the jellyfish. And these 2 systems are very much apart evolutionarily. So the yolk sac, the avian yolk sac, is essentially a system from a bilaterian animal, an animal vertebrate, bilaterian, right? And its very, very distant relative is the jellyfish. So I'm going to start with discussing the first system, the avian yolk sac, which actually has a heart. Now, here I'm returning to the yolk sac. You see the heart beating. This is the heart of the embryo, and that heart pushes fluxes through the vascularized yolk sac, which essentially will feed the embryo, right? So the heart establishes flows, and here are some length scales related to the problem. Now, we wanted to study the dynamics of those fluxes, and the data came from Liz Jones, and basically those data consisted of her injecting fluorescent nanobeads into the system, and then in a lawnmower fashion, as you see in that picture, in a lawnmower fashion, tiling essentially the system, acquiring movies of about 2 seconds, maybe 6 or so heartbeats. Now, the big problem here is that the system is alive. Unfortunately, in biology, things tend to be alive. And as we are progressing in this lawnmower fashion, between the tiles, there is shifting. There is slight remodeling of the vessels, and so the heartbeat also can change, right? So we had a lot of work to do in order to stitch it in a consistent fashion, but I won't talk about it. Now, in parallel, we were working in developing the theory on how to model in a very computationally efficient manner the pulsatility in the flows, basically the transmission of that pulse. Now, what you see on the left column is the measurements as we extracted them from the data. The top row is the mean flow, and the second row is the pulsatility index, basically how strongly pulsatile the flow is. And you see between measurements, left column, and simulation, right column, we have relatively good agreement. I will not tell you where the bodies are buried. Now, what you can notice is that the pulsatility index, how pulsatile the flow is, at least in this particular stage, cannot reliably determine arteriovenous identity. Basically, on the left side of this embryo as displayed here, you would be expecting more of a venous identity, and on the right side, more of an arterial identity. But as you see, it's kind of the same. This goes against several papers in the literature which says pulsatility is the marker. Now, while we were, in order to understand what was going on, we did a lot of tool development that allowed us to navigate the whole organ and basically integrate theory, computation, and data to find the vascular architecture, pressure, the flow waveforms, the relative phase of the transmitted pulse, for the whole organ, as you will see. Now, why did we do that? Because we did that because we wanted to understand at the vessel level what determines remodeling, the increase of the lumen diameter. Now, the paradigm, the proposed computational models in the literature does not include short-term dynamics at all, the pulsatile waveform, but we know from molecular and cellular data that dynamics matters, right? So we wanted to measure that dynamics, and this is what I'm going to be focusing on this part of the talk. So the easiest thing to do first is to look at the spectral content of the pulse. The venous side you see on the left, and the arterial side you see on the right here displayed. And the first thing that you can notice is that the spectral content between the two sides is very, very different. And then what we did is that we went and we looked at the arteriovenous similarity of the pulse, basically the measured pulse at each little vessel, how similar it is to the prototypical venous and the prototypical arterial. And you see that the arteriovenous content is balanced. And if the vessels could actually measure that, that would give us the right content, essentially, of arteries and veins. Another interesting thing that we saw is that the wavelength, here you see it displayed here on the top right, you see the phase. The wavelength is about the system size, which is interesting. We don't know how to think about it yet. Other things we could do is, of course, we could do digital twins here, and we could see, predict what would happen in atherosclerotic embryo. We can do that computationally, and then we see if the vessels are stiffer, the pulsatility goes up everywhere, and then the arteriovenous similarity changes, and everything ends up should end up looking like a vein. On the contrary, if I make the vessels more compliant, softer, basically, the pulsatility goes down, and the arteriovenous similarity becomes more like an artery. The arterial identity would dominate. We haven't done these experiments, but these are predictions. Other things we could do is that we did ligate computationally the right dorsal aorta and calculate non-local effects. For example, on the top right of the embryo, we see in the ligated case, we would predict loss of arterial identity, and in the normal case, we would start building arteries then. Now I'm going to switch to the second system. I'm going to switch to the jellyfish now, which, unlike the yolk sac, there is no centralized heart. In fact, as I will try to convince you, the whole movement of the animal acts a little bit like a heartbeat. Now, this is a primer on the jellyfish. Now, this is if you take a jellyfish and you put it basically the umbrella, you put it basically upside down on a Petri dish, what you would be seeing is the picture on the left. In the center, there is basically there's a hole, which is the mouth. And that is both input and output for food, and I will let you guess what this means. Now, the food, seawater with food comes in, goes through the stomachs that you see, the four lobe things over there, and then it goes, according to the single very old paper where it was described, it goes to the perimeter through the straight canals, get dispersed on the circular canal, and gets collected by the branch tree that you see and brought back and expelled, right? Now, we didn't know a priori, these were all descriptions, we didn't know what drives the flow. So we, and by we I mean anemic, did experiments, and cilia, the tubes, the inside of the tubes are ciliated, there are metachronal waves. There was a poster yesterday that are the primary, let's say, deciders of the directionality of the flow. That we can test if we anesthetize a jellyfish and actually record the fluxes. You see on the left, mostly in the basically unbranched canals, it's unidirectional, but also, okay, my slides are a bit of a disaster. Okay, that movie didn't play. Anyway, but they could be also very, very complex. Now, this is an awake jellyfish. You see it here pulsing. And also what you can see is that the dimensions of the vessel change dramatically, not only the radius, but also the length. So what is described mathematically in the literature, so the mathematics that are developed, is radial peristalsis, the radius of a vessel or a tube changing and how that contributes to flow, but not longitudinal. So we went and we did the math for that. And the slides are displaying very, very badly, although we tested them, so I apologize. So we developed the mathematics, let's say, for longitudinal peristalsis, and we could actually calculate where essentially the particles would go under these peristaltic waves. Then we could put together networks of canals like an idealized jellyfish and predict where the fluxes would be going, and this is now in preparation. Now, what we needed to do, though, is also look at actually real jellyfish, in this particular case in the Petri dish. We're tracking the canals, we're tracking the deformations, and in this particular case, I want to show you a little bit what the deformation looks like. Now, the typical contraction lasts maybe half a second up to 0.7 seconds, and it's mostly synchronous. It's not exactly synchronous, it's not radially symmetric. So now we went and we tracked both the deformation of the vessel, and on the right you see the same vessel simultaneously computationally stabilized, and then the trajectories of the particles were tracked. And then we put this thing together. Now, what you see on the right, on the top, is the velocity of the particles, of the flow, basically, along the longitudinal axis of the vessel, basically how fast the particles are going. On average, it's roughly about 260 microns per second. And in this particular case, the animal is anesthetized. This is just what the cilia would be doing by themselves. Now, what we went and we did now is we looked at it in the awake animal, and what you see in the plot is that you see these massive contractions that would have been marked with long red bars that don't show up here, but you can see the contractions happening. And that the contractions really essentially cause the flow to shoot. I should say they perturb the velocity of the flow. There's nothing surprising there. Now, there is something that should give you huge pause here, actually. If you're a fluid dynamicist in the audience, these are low Reynolds number flows. These are basically Stokes flows, which means what this translates to is that when the contraction is done, the flow should immediately revert to what we would have by, by the cilia alone. But this is not what's happening. There is a relaxation time scale, which is about a second, where the particles remain accelerated. And what this means practically is that we believe, what this means practically is that there is synergy between the contractions and the cilia beating. So either two mechanisms would be okay, but together they are really, really good. Now it's in progress to, we're trying to do freely moving jellyfish, basically quantify the contractions there, but this is in progress. I'm not going to talk to you about. This would have been the summary slide, but you can't see anything. Now, I want to finish with a story, a little story of a trip to an aquarium I took with my son, and he was absolutely fascinated and terrified by the sharks. I was fascinated and terrified by something else, the jellyfish. And then I was walking around, I saw something that I hadn't seen before, and that is the upside-down jellyfish. It's basically, it's a species of jellyfish that it's actually quite well studied, that drops to the ocean floor and keeps pulsing though, so I was very puzzled why it keeps pulsing. And I read there is some ideas that it's doing it essentially to promote exchange of water around the body of the oral arms, but in the spirit of HFSP, I tend to be a little bit more critical about these things, and maybe in the spirit of HFSP, I will tend to be more, let's say, accepting of a little bit crazy ideas that maybe this also helps its digestion. So with that, I would like to help the rest of the consortium, the HFSP funded consortium, my group, and you for your attention.
Thank you very much.